Hello everyone, I'm Kate da Silva and this is my presentation on 3D printed ovarian molds. The objective was to create a mold for ovaries to prepare consistent histological slices. The idea is that an ovary can be placed and manipulated to achieve regular size samples for further investigation. I'll be going over ovarian function and anatomy, Dr. O'Neill's sectioning scheme, the analysis of the tissue, and the 3D print mold itself. This was part of the Pixel Lab, which is a Penn image computing and science laboratory. My PIs were Dr. Allison Pouch and Dr. Jim G. My co-PI was Dr. Kim Jung-Yu. The graduate student that helped a lot was Olivia Sandvold, and collaborators were Dr. Kate O'Neill and Addie Johnston. Ovarian function, ovaries are the primary reproductive organs with three important functions. They protect the eggs of females born with, they release eggs for potential fertilization, and secrete hormones from the corpus luteum. The ovaries release one egg, sometimes more, each menstrual cycle through ovulation, and each ovary contains follicles, and inside each follicle is a dormant egg. Before puberty, ovaries are just long bundles of tissue, and when they mature, they become to be about the size of an almond, but sizes can vary between females. Typically, a female is born with two ovaries, and they contain around 150,000 to 500,000 follicles. And I found it interesting that by sexual maturity, these drop to about 34,000 follicles. When an egg is stimulated by hormones to maturity, the follicle migrates to the ovary wall, and the egg and follicle grow and mature. And these mature follicles are known as Graafian follicles, which can grow up to 1.2 inches or 30 millimeters in diameter. The follicle contains a fluid that protects the egg as it grows, and when the egg is released, it bursts, releasing the egg to the nearest fallopian tube. From the fallopian tube, the egg travels to the uterus, and if it's not fertilized, then it's flushed out of the uterus through menstruation, which is a cycle that lasts 28 to 32 days. The anatomy, the tunica albuginea is a condensed layer of fibrous tissue on the outside of the surface of the ovary. The uteroovarian ligament, or the ovarian ligament, is a fibromuscular band that spans from the uterus into the broad ligament. And the infundibulopelvic ligament, or the IP ligament, also known as the suspensory ligament, is what attaches the extreme lateral part of the fallopian tube to the pelvic sidewall. The mesovarium is what suspends the ovary, and it's also where the cut is made in order to do analysis of ovaries. The cortex is the outermost layer of the ovary, composed of stroma and developing follicles, while the medulla is the middle portion of the ovary, composed of stroma, blood vessels, and nerves. And the hilum is what supplies the blood vessels and nerves into the functional tissue. Dr. O'Neill's sectioning scheme can be seen to the right. First, you start with the whole ovary. Next, it is cut into medial, central, and lateral poles. And originally, she was thinking about doing between 6 to 9 sections, but she decided she wanted 4 sections per pole for a total of 12 sections. Next, each of these sections it's very hard to see where the cortex ends and the medulla starts. Each section is placed in formalin, and then quarter slices of the sections are created by taking it laterally and cutting them into quarters in sort of a clockwise motion. Then each piece is put through a process called snap freezing, which is the rapid cooling of the tissue for preservation. Next, it's put through OCT, which is optimal cutting temperature compound, as seen here. And this is to mount very thin slices, about 0.5 millimeters, onto slides. From there, the pathologist stains the slides with H&E to determine the normal morphology of the tissue. Then RNA is extracted from the tissue and sequenced and matched within the region, and the localization of cells that takes place is from 3 to 4 cells per region. Originally, Dr. O'Neill wanted to use the Thomas Sadie Riggs tissue slicer, as seen here, but she found that there was some position difficulties, so instead she opted for freehand cutting, and she explained that it was much like cutting an onion. So other disadvantages to the Thomas Sadie Riggs tissue slicer is that the ovary has difficulty laying flat since it's circular or oval shape. You can't adequately view the sections to be cut. There's also size difficulties because it varies from patient to patient, and position difficulties, as mentioned before. So the alternative was to create 3D printed molds customized to ovary side to aid in consistent cross-sectioning for subsequent analysis. So I created the 3D print mold using Fusion 360, which is a computer automated design program that combines shape modeling with mechanical design and manufacturing for product design and development. Python is what I use to automate the mold, as seen here, and you can view the mold running itself. So then for the mold itself, ABS, which is acrylonitrile butadiene styrene, was used, which is a hard plastic, instead of PLA, which is polyactinide, which is another type of plastic. When we printed the PLA mold, we found that the sections right here would fold in on themselves, so it wasn't very sturdy. For the code itself, open source code was used. So ABS was also chosen because it can withstand temperatures from -10 degrees Celsius to 100 degrees Celsius. And the specifications for the mold itself, it creates 12 uniform sections, and each section here is about 0.5 millimeters for the blade. So an advantage was that the ovary lays flat. The sections can be adequately viewed to cut. It also created uniform cuts, and it was faster slicing. So a disadvantage was that ABS printed parts can't go through autoclave due to the heat, so it has to be hand washed. When the mold was tested, shedding of the material was found with slicing, but this can be mitigated by increasing the size of the blade sections. And there's poor conductivity with the mold itself, so this wasn't a problem with this particular project because they were doing the cuts at room temperature. But if anyone ever wants to take the mold and the ovary and keep them together and have them both frozen, you would need a different material. So to the right, you can see the mold in action, and this is it cutting the ovary. I swear it's not chicken breast. So I'd like to thank my PIs, Dr. Allison Pouch and Jim G. I'd also like to thank the graduate student, Olivia Sandvold, who helped with the animations on this project, and Casey Henson for being there to let me know what looked good and what looked terrible about my project. I'd like to thank FJ for the help with Python coding, and I'd like to thank Dr. O'Neill and Addie for the tissue samples and for the images, and thank you everyone for listening to my presentation, and HuBMAP and the NIH for this opportunity.